thank you for Covaris for this opportunity and for your kind invitation to be here at ASMS. And yeah, today I would like to walk you through our how we use Covaris CryoPREP and also the AFA technology for our metabolite and lipid extraction, and that as an example for the analysis of cancer tissue. Allow me first to introduce our group. So we are part of the University of Tubingen in Germany. And a couple of years ago, it was established this new research center called M3, which is in short for malignome, metabolome, and microbiome. And within this center, it's our group, the core facility Metabolomics. And at the same time, it's also a research center, a research group, and both are led by Dr. Christoph Trautwein. Here are a few members of my team who made it into the website, but actually the team is pretty much bigger. And then, yeah, we are equipped with state-of-the-art instrumentation for targeted, untargeted, and quantitative, and as well also for spatial metabolomics and lipidomics. So to set the stage, why focus on metabolomics and lipidomics? To mention just a few reasons, and these fields sit at the bottom of the omics cascade, and they reflect the real-time cellular changes which are shaped by gene expression and environment, and also they are key for understanding biochemical changes in health and disease. They are very helpful to discover biomarkers, metabolic pathways mapping, drug mechanisms, also relevant in studying and monitoring diseases and treatments. And then specifically to lipids, they play key roles in membrane structure, signaling, and also in energy storage. But due to the complexity and diversity of these molecules, we need high sensitive and reproducible workflows to be able to analyze them. And especially when working with solid samples like tissues. So to capture the real-time cellular changes, the sample preparation becomes critical. Here is just an example of a basic metabolomics workflow. And what I want to actually highlight is that we are working with many different sample types. We have urine, for example, or plasma, which are more straightforward to extract, as metabolites are more accessible. But on the other hand, we have tissues and cells with stronger membranes, like organoids and bacterial cells that are more complex. So for that, you need a few key considerations. Would be that you need to fully disrupt the cells to release all the metabolites. You need to avoid temperature to not degrade your metabolites. Then also avoid sample loss and cross-contamination. And of course, ensure reproducibility so that the metabolite changes reflect really the true biological differences. And then, of course, if you have many samples, it would be good to have some scalability and higher throughput. So many conventional methods fall short on some of these key considerations. And let me show you how we have addressed these challenges using the Covaris technology. So here's an overview of our first step, which is pulverization. We use frozen tissues, mostly from rat or mice, and in this case, brain, but also, of course, liver and kidney. Then we insert them into the tissue tube, and this goes into the CryoPREP, where the standardized impact energy ensures us that there is consistent and complete pulverization with minimized temperature generation. And that's good because you can choose from several impact steps. And then, thanks to the screw-on connection between the tissue tube and the millitube, you can transfer the sample nicely, avoiding losing the sample or contaminating it. And then on the other hand, for cells or organoids, as I was saying, we skip the cryoprep and we put them directly into the millitubes. And after that, we do either two-phase extraction if we want to separate into a polar phase to analyze the metabolomics, and a polar phase for the lipidomics. And if you want to have everything together, then we just do the one-phase extraction. So after adding the appropriate solvents, the next step is ultrasonication with the LE220 plus. And samples are inserted into the tray. We can insert up to 24 samples. And then it runs 4 in parallel. And this allows us for higher throughput processing and also with minimal hands-on time. And going to the ultrasonicator, this provides efficient, cold at minimum 6 degrees temperature, degrees Celsius, and contact-free extraction, and that with very precise control over several parameters that you can optimize. And these are key factors for consistency across the samples. So once the sonication is complete, which for a method it's about 3 minutes per row, we then centrifuge, dry, and reconstitute the samples for LC-MS analysis. First of all, to assess the sample degradation during handling and extraction, in collaboration with our NMR group, which is also led by Dr. Trautwein, we performed some complementary quality control. For that, we calculated the adenylate energy charge. Although we found ATP in all the samples, the value was a bit low, which indicated some degradation. We assumed to be rather in the pre-extraction handling. But because we found ATP that also ascorbate and reduced glutathione in all the samples, and that indicates that our extraction workflow preserved the redox-sensitive metabolites for further breakdown. Moving on to the analysis setup, we have dedicated columns for reverse phase for the nonpolar metabolites, for HILIC also for the polar metabolites, and then also more dedicated work method for lipidomics. Use our timsTOF-MS with a VIB hazy source for increased sensitivity. And then the acquisition mode in the MS was the passive and with TIMS stepping. For the annotation of metabolites, we use both internal standard libraries, but we have like 1,100 metabolites to attain the highest level of confidence. And then we also have purchased or public target lists and spectral libraries, and also a prediction tool, which is called the rule-based lipid species. And the identifiers we use for confidence in our annotations are mass to charge ratio, less than 2 ppm. We have the isotopic fidelity, less than 20, the MSMS score, more than 900, and the collision cross-section, less than 1% deviation. And of course, we also have, for quality control, QC samples, linearity series, and blanks. Going now to more specific example for our untargeted metabolomics, I'll show you some results from the study of the mouse brain. We use 10 to 20 milligrams of fresh frozen biopsy, and then we are comparing a control group versus one that was treated. As this data is not published yet, I will not dig very deep into the details, but I'll just show you a few examples. Let me start by showing the coverage we obtained, running both reverse phase and HILIC. I have to say that numbers are a bit low, and that's because we didn't include yet our internal library. But you can see for the HILIC positive, we had 121 annotations. We increased it to 186 with the internal library. So probably these numbers will be a bit higher. But anyways, we got exclusively 258 metabolites, and also additional with the NMR, 80 more. Going now at metabolite level, I'd like to show you this heat map where it's compared the control group in the red, the left, and then the green is the treated one. And the blue rectangle, you see the metabolites that decreased in concentration, comparing the two, and in red, the ones that increased. And these are only the significant ones. So one interesting example is the N-acetyl neuraminic acid, which decreased, and that is linked to immune exhaustion. So we can say that a lower level may indicate that immune recovery. Another example could be the N-acetyl aspartic acid, which is often considered a marker for neuronal viability and density, and a higher level of this metabolite would indicate a lower tumor impact. Then using the two-phase extraction that I showed you earlier, we got one of the phases for the lipids, and we run only positive ionization mode. We annotated here 264 lipids across 18 lipid subclasses. Here's just an abundance pie. You can see that the highest one was the TGs and the Ps, PS, and so on. But to show you a bit more in detail, here are two that were significant. Also, like before, comparing the control and treatment, we see increase in these two lipids from PEO group and TG, and that indicates some alteration in membrane dynamics, lipid signaling, and also oxidative stress. So a few thoughts on our conclusions would be that different biofluids, tissue samples, and similarly complex matrices require a more dedicated pre-analytical workflow prior metabolomics. And metabolites and lipids should be properly extracted, conserved, not degraded, with high recovery. And our Covaris-based workflow, combining the cryogenic disruption and AFA ultrasonications, offers a robust, scalable, and reproducible platform for extracting these metabolites and lipids, and of course also from complex tissues, and that ensuring high data quality, comparability, and of course biological insight, which is the most important part. And then we are planning for further optimization in these extraction parameters that I was showing you, and focused on to obtain maybe if MS cleaner samples. So we'll be focused on that in the future, and with Covaris help as well, I think. Here are a few of our publications with the Covaris workflow, and many more on the way with different samples. And finally, I would like to thank you for your attention and also for the invitation to be here.